Hello, good morning. My name's Louis Sparling and I am working as a research pharmacist. Today I would like to explain in this presentation how modern drug research and development works from the discovery of potential drug molecules through to the design and testing of new medicines. As pharmaceutical managers yourselves, many of you come from backgrounds of business, finance, operations, and project management rather than chemistry of pharmacology. Therefore, I will explain the scientific concepts clearly and demonstrate why continued investment in pharmaceutical research is essential for future innovation. During this presentation, I will discuss what drugs and medicines are, the important pharmacological concepts such as placebo effects and therapeutic windows, how molecular structure affects drug action, how new drug candidates are identified, and the increasingly important role of computer technology in drug design. My aim is not only to educate, but also to demonstrate why investment in research remains one of the most important drivers of healthcare innovation and commercial success. This slide outlines the outcomes that will be met in this presentation. Please also refer to the handouts provided, which summarize the slides in this presentation, and I will take any questions at the end. Before discussing drug development, it is important to define what we mean by the terms drug and medicine. A drug is any chemical substance that provides a biological effect when introduced into the body. Drugs work by interacting with cells, receptors, enzymes, or biochemical pathways. A medicine is a drug that has been tested, approved, and used for the prevention, diagnosis, or treatment of disease. Medicines can produce a wide range of beneficial effects. For example, antibiotics kill bacteria, insulin regulates blood glucose levels, and painkillers reduce pain and inflammation. Oh, sorry. However, because drugs alter normal biological processes, they can also produce unwanted effects. Therefore, one of the major goals of pharmaceutical research is to maximize therapeutic benefit while minimizing risks to patients. The effectiveness of a medicine depends on factors such as dose, route of administration, patient characteristics, and molecular structure. One important concept in clinical research is the placebo effect. A placebo is a substance that contains no active therapeutic ingredient but may still produce an apparent improvement in symptoms. Examples like the one in this slide here are that sugar tablets can be used as a placebo. The placebo effect occurs because a person's expectations can influence how they perceive such pain, fatigue, and anxiety. For example, if a patient believes they are receiving effective treatment, measurable improvements may occur even though the treatment contains no active drug. This is why placebo-controlled clinical trials are considered the gold standard in pharmaceutical research. By comparing patients receiving the active drug with receiving a placebo, researchers can determine whether medicine genuinely produces therapeutic effects beyond psychological expectation. Without placebo-controlled studies, it would be very difficult to accurately evaluate the effectiveness of new medicines. Another important pharmacological concept is the therapeutic window. The therapeutic window refers to the range of drug concentrations between the minimum effective concentration and the concentration at which toxicity begins to occur. If the concentration is below the therapeutic window, seen on the scroll, the medicine may not provide sufficient benefit. If the concentration rises above the therapeutic window, harmful side effects or toxic effects may occur. Some medicines have very wide therapeutic windows and are genuinely safe. Others have a narrow therapeutic window and require careful monitoring. Examples of drugs with narrow therapeutic windows include warfarin, lithium, and digoxin. A major objective during drug Major objective during drug development is to create medicines with wider therapeutic windows because this improves patient safety and reduces the likelihood of accidental overdose. We now move on to tolerance. Tolerance develops when repeated exposure to a drug reduces its effectiveness over time. As tolerance develops, higher doses may be required to achieve the same therapeutic effect. There are several mechanisms by which tolerance occurs. Firstly, receptors may become less sensitive to stimulation. Secondly, the number of receptors may decrease. Thirdly, the body may become more efficient at metabolizing and eliminating the drug. Examples of drugs commonly associated with tolerance include opioids, nicotines, and benzodiazepines. Tolerance is an important consideration during drug development because it can affect long-term effectiveness, patient safety, and the risk of dependence. Researchers therefore aim to develop medicines that maintain effectiveness while reducing the likelihood of tolerance developing. We now move on to side effects. Side effects are the unintended effects that occur in addition to the desired therapeutic effect. Some side effects are relatively minor and manageable. In this slide, I have demonstrated the possible side effects of taking antidepressants. This list is from the NHS website on information for patients on antidepressants and their side effects. Some side effects can be serious and potentially life-threatening. For example, with antidepressants, overdose can cause serotonin syndrome or even death. This is if the prescribed dose is exceeded by the patient. One major goal of medicinal chemistry is to improve drug selectivity so that a medicine interacts primarily with the desired receptor or enzyme. Improved selectivity generally leads to fewer side effects and better patient outcomes. We now move on to chemical structure and drug function. Drug molecules exert these effects because of their chemical structure. The shape, size, and arrangement of atoms within a molecule determine how it interacts with biological targets. The relationship between molecular structures can significantly alter effectiveness, safety, and duration of action. Medicinal chemists use SAR studies to understand which structural features are essential for activity and which can be modified to improve performance. Understanding structure-activity relationships forms the foundation of rational drug design. We now move on to similarities and differences in related drug molecules. Many drugs belong to families of chemical-related compounds. A useful example, like the one here on this slide, is morphine, codeine, methadone, and heroin. These drugs share a similar molecular backbone but differ slightly in structure. Despite these similarities, their biological effects differ substantially. Morphine is a powerful analgesic. Codeine is less potent and is often used for mild to moderate pain. Heroin crosses the blood-brain barrier more rapidly and produces stronger euphoric effects. Methadone is used as a prescription medication to help with withdrawal from previous heroin use. Small structural modifications therefore have major consequences for pharmacological activity. This illustrates why medicinal chemists carefully study structural similarities and differences during drug development. Now, functional groups are specific arrangements of atoms within molecules that determine chemical behavior. Common functional groups include alcohols, amines, carboxylic acids, esters, and amides. These groups influence many important properties, including solubility, polarity, stability, and receptor binding. Because functional groups have such a strong influence on drug behavior, they are frequently modified during medicinal chemistry programs. The ability to manipulate functional groups allows researchers to optimize drug performance and improve patient outcomes. Functional group modifications like the ones in this slide, can increase the effectiveness of medicines. For example, introducing a functional group that improves receptor binding may increase potency. Greater potency means smaller doses can achieve the same therapeutic effect. This can reduce manufacturing costs and minimize side effects. Medicinal chemists often synthesize hundreds or even thousands of related molecules before identifying the most effective compound. The optimization process plays a crucial role in modern pharmaceutical development. Now, improving drug delivery and safety. Functional group modifications can also improve drug delivery and safety. Adding polar functional groups may increase water solubility and improve absorption. Increasing lipid solubility may improve movement across cell membranes. Some modifications create prodrugs. A prodrug is an inactive compound that becomes active after metabolism within the body. Codeine is a well-known example because it is converted into morphine after administration. Prodrugs can improve absorption, reduce toxicity, and enhance compliance. As a result, functional group modification remains one of the most valuable tools available to medicinal chemists. Now, we move on to geometrical isomerism. Some compounds exist as geometrical isomers. Geometrical isomerism occurs when molecules contain the same atoms but differ in their spatial arrangement. The two forms are known as cis and trans isomers. In cis isomers, similar groups are located on the same side of a double bond. In trans isomers, similar groups are located on opposite sides. Although these compounds possess the same molecular formula, their three-dimensional shapes differ significantly. Because biological receptors are highly shape-specific, geometrical isomerism can have important pharmacological consequences. So, a famous example is cisplain. Cisplain is a platinum-containing anti-cancer drug used to treat several forms of cancer. Its success depends on the cis arrangement of ligands around the platinum atom. The trans form, known as transplain, has dramatically reduced anti-cancer activity. This demonstrates that biological activity depends not only on the atoms present, but also on their three-dimensional arrangement. Understanding geometrical isomerism is therefore essential in pharmaceutical chemistry. I'll move on to slide 15. Chirality is another important concept. A molecule is chiral if it cannot be superimposed on its mirror image. The easiest way to visualize this is considering human hands. Both hands contain the same compounds but are mirror images that cannot be perfectly aligned. Chiral molecules exist as two mirror image forms called enantiomers. Although they share identical chemical formulas, their biological effects may differ substantially. Optical isomerism occurs because biological systems are themselves chiral. Receptors, enzymes, and proteins can distinguish between different enantiomers. Consequently, one enantiomer may be highly beneficial while the other may be less active or even harmful. One example is ibuprofen, where there is one enantiomer that provides significant therapeutic activity. An even more important example is thalidomide. One enantiomer produced useful therapeutic effects, whereas the other was associated with severe birth defects. The tragedy highlighted the importance of stereochemistry in pharmaceutical development. Today, many modern medicines are produced as single enantiomer products to improve effectiveness and safety. And the thalidomide, problems with the thalidomide were mainly birth defects, sort of as not properly deformed limbs. Techniques used for drug detection. Before a medicine can be improved, scientists must be able to accurately detect and measure it within biological samples such as blood, urine, saliva, and body tissues. Drug detection is important for several reasons. It allows researchers to monitor how much drug enters the bloodstream, also determining how quickly it is eliminated from the body, investigate potential toxicity, and ensure patient safety. Drug detection is also used in forensic investigations, anti-doping testing in sport, and monitoring compliance with prescribed medication. Because drug concentrations are often extremely low, highly sensitive analytical techniques are required. These techniques must be capable of identifying specific molecules within complex biological mixtures containing thousands of different compounds. The most commonly used techniques include chromatography, spectrometry, and mass spectrometry. These methods provide accurate and reliable information regarding the identity, concentration, and behavior of drugs within the body. Understanding how drugs are detected is essential because accurate measurement forms the foundation of pharmaceutical research, clinical trials, and therapeutic monitoring. One of the most widely used analytical methods is chromatography. Chromatography works by separating compounds within a mixture according to differences in their chemical properties. Different compounds travel through a stationary phase at different rates, allowing them to be separated and identified. Examples include gas chromatography, which is often used for volatile compounds with extremely high accuracy. Spectroscopy is also widely used. These techniques are used to examine how molecules interact with electromagnetic radiation. Examples include UV visible spectroscopy, infrared spectroscopy, and nuclear magnetic resonance spectroscopy. These techniques are often combined to provide both separation and identification, making it one of the most powerful tools in modern pharmaceutical research. Now move on. The discovery of new medicines begins with identifying molecules that may influence a disease process. Historically, many medicines were discovered by chance. However, modern pharmaceutical research uses systemic approaches to identify promising drug candidates. One important method is natural product discovery. Many successful medicines originate from plants, fungi, or microorganisms. Examples include penicillin, which was discovered from mold, and another example is paclitaxel, which is an important anti-cancer drug originally isolated from the Pacific yew tree. Researchers also investigate biological pathways involved in disease. By understanding the molecular mechanisms responsible for illness, scientists can identify proteins, enzymes, or receptors that may serve as drug targets. Once a target has been identified, compounds can be screened to determine whether they interact with it effectively. The identification of suitable lead compounds is one of the most important stages in drug development because it provides the foundation for future optimization. One of the most important modern discovery techniques in high throughput screening is high High throughput screening uses automated robotic systems capable of testing hundreds of thousands of compounds against a biological target in a relatively short period of time. The objective is to identify compounds that demonstrate promising activity. These compounds are known as lead compounds. Once identified, lead compounds undergo optimization to improve potency, selectivity, and safety. Another increasingly important strategy is drug repurposing. Drug repurposing involves finding new uses for medicines that have already been approved for other conditions. This approach offers several advantages because the safety of these medicines are already well understood. An example is sildenafil, which was originally investigated for the treatment of angina, but later became widely used for erectile dysfunction. Drug repurposing can significantly reduce development costs and accelerate the delivery of treatments to patients. We now move on to computers in drug design. Computers have transformed pharmaceutical research and development. Traditionally, drug discovery relied heavily on laboratory experiments. While laboratory work remains essential, modern computer techniques allow researchers to predict molecular behavior before compounds are synthesized. Computer-aided drug design enables scientists to visualize molecules in 3 dimensions and predict how they might interact with biological targets. This reduces the number of compounds that need to be synthesized and tested experimentally. The use of computers in drug design saves time, reduces costs, and improves efficiency throughout the drug development process, which is good. The use of computer technology has become particularly important because pharmaceutical development is expensive, often, as we all know, often costing billions of pounds and taking more than 10 years from discovery to market approval. By improving efficiency, computer-aided methods help reduce both financial risk and development timelines. So one important computer-aided technique is modular docking. Modular docking uses computer simulations to predict how well a drug molecule fits into the binding state of a biological target, such as an enzyme or receptor. Scientists can estimate the strength of these interactions and identify compounds with the greatest potential for therapeutic activity. Artificial intelligence and machine learning are now becoming increasingly important in pharmaceutical research. These systems analyze enormous data sets and identify patterns that may not be obvious to researchers. AI can predict biological activity, identify side effects, and optimize molecular structures. Recent advances in artificial intelligence, or AI, have accelerated research such as cancer treatment, antibiotic discovery, and personalized medicine. So we're coming to the end now. So in conclusion, the discovery and development of medicines is a highly complex process that combines chemistry, biology, pharmacology, and computer science. Today, I have explained what drugs and medicines are, the concepts of placebo, therapeutic window, tolerance, and side effects. Also, how chemical structure influences drug behavior, the importance of functional group modification, geometrical isomerism, and optical isomerism. I have also talked about the techniques used for drug detection, methods used to identify new drugs, and the growing role of computers and artificial intelligence in drug design. These advances allow us to develop safer, more effective, and more targeted therapies. Continued investment in pharmaceutical research is therefore essential for improving patient outcomes, advancing healthcare, and maintaining competitiveness within the pharmaceutical industry. Every successful medicine begins with investment in research. Continued funding allows researchers to discover safer treatments, reduce healthcare costs, and improve patient outcomes worldwide. I hope you have enjoyed this presentation and that you can help us by providing additional funding for pharmaceutical developments. Thank you for listening. I have also included a list of the references used in this presentation and also a list of the images so you can click on the links if you need to. And please refer to your handouts, and thank you very much for listening. Thank you.